Hello dear viewers, welcome to Mr.Zoologist YouTube channel. Myself Nasir, and today we are going to discuss about CDK inhibitors or cyclin dependent inhibitors. The main objectives of this course are to get knowledge and about cancerous cells, structure and function, understanding biology and genetics of cancerous tissue, learn the effect of various chemical carcinogens on different tissues of animals. So these are the different phases of the cell cycle, such as the G1 phase, S phase, G2 phase, M phase, and GO phase, resting phase. So what are CDK inhibitors? The cell cycle progression is also regulated by a class of proteins called CDK inhibitor or simply CDKI. Seven CDKI have reported to date, such as P16 INK4A, P15 INK4B, P18 INK4C, P19 INK4D, P21 CYP1, P27 KIP1, and P57 KIP2. So, cyclin CDK inhibitors, the first four inhibitors are grouped as INK4 proteins and target CDK4/6 complexes. They have no effect on CDC2 and CDK2. The remaining three inhibitors are more widely acting and target inhibit the other cyclin CDK complexes throughout the remainder of the cell cycle, such as that you can see that in the INK4 family actually, that includes 16 INK4A, 15 INK4B, and 18 INK4C, and 19 INK4D. These all inhibit the D-CDK4/6 complexes, D-cyclin CDK4/6 complexes. Whereas P57 KIP2, P27 KIP1, and P21 CYP1 block the passage throughout the remainder of the cell cycle, such as HCDK2, ACDK2, ACDC2, and BCDC2. And if we talk about the functions of the INK4 proteins, the CDK inhibitors block the actions of CDKs at various points in the cell cycle. The four INK4 proteins, P16 INK4A, P15 INK4B, and P18 INK4C, and P19 INK4D, are specialized to inhibit the D-CDK4 and D-CDK6 complexes that are active in early and mid G1 phase. So if we talk about the function of CYP CKIs, the three CYP CKIs, P21 CYP1, P27 KIP1, and P57 KIP2, can inhibit the remaining cyclin CDK complexes that are active throughout the cell cycle. Under certain conditions, signaling containing within the cell, such as signals indicating damage to the cell's DNA, can halt advance through the cell cycle by blocking the action of cyclin CDK complexes that are activated in the S and G2 phase of the cell cycle. Paradoxically, oppositely, P21 CYP1 and P27 KIP1 are known to promote formation of DCDK4/6 complexes during the G1 phase. So this depiction illustrates that how one domain of P27 KIP1, light green in color, blocks cyclin A CDK2 function by obstructing the ATP bonding site in the catalytic left of the CDK enzyme. And that is actually responsible for the blocking of the CDK, D-CDK or CDK2 complex, blocking its phosphorylation targets. So in P16 INK4A shows here brown in color, bind to CDK6, reddish in color, and to DCDK4. Not shown, these CDK inhibitors distort the cyclin binding site of CDK6, reducing its affinity for D-type cyclins. At the same time, they disrupt the ATP binding site and thereby compromise catalytic activity. So the action of these two classes of CDK inhibitors are shown by P15 INK4B and P21 CYP1 when TGF-beta cytokine to enhance the cell growth and are produced by all white blood cell lineages is applied to the epithelial cell that shows the number of downstream responses that opposes cell proliferation. So among these are significant increases in the levels of P15 INK4B, which proceeds to block the formation of cyclin D-CDK4/6 complexes and to inhibit those that have already formed. Without active D-CDK4/6 complexes, the cell is unable to advance through the early and mid G1 and reach the R point. Once the cell has passed through the R point, the action of the D-CDK4/6 complexes seem to become unnecessary. So P21 CYP1 are more widely using as acting CDK inhibitor is also induced by TGF-beta, but very weakly. It is also induced by damage to the cell's genome. Once a cell is present at significant levels, P21 CYP1 can act throughout the much of the cell cycle to stop a cell in its tracks. As long as the genomic DNA remains in an unrepaired state, the P21 CYP1 that has been induced will shut down the activity of the already formed cyclin CDK complexes such as ECDK2, ACDK2, ACDC2, and BCDC2. So if a cell's genome becomes damaged during G1 due to the actions of the various mutagenic agents such as P21 CYP1 will block the advance of the R point by inhibiting the ECDK2 complexes until the damage has been repaired, ensuring that the cell does not progress into the S phase. In addition, P21 CYP1 can inhibit the functions of a key component of the cell's DNA replication apparatus termed pCNA, proliferating cell nuclear antigen. This ensures that already initiated DNA synthesis is halted or stopped until the DNA repair has been completed by the DNA repairing pathways or mechanisms. So these are the diagrams showing the actions of the INK4 protein family and the CIP and KIP family that is all occurring inside the cell, because the genetic material is present inside the cell. The cell is dividing. First of all, if we talk about the cell cycle, in the cell cycle, in the process of the mitosis or meiosis, first of all is a process that occurring is kinases. To check it, there are different levels of the cyclins and the CDKs and inhibitors. So control of cell cycle progression by TGF-beta. TGF-beta controls the cell cycle machinery in part through its ability to modulate the levels of CDK inhibitors. It acts to strongly induce increased expression of P15 INK4B and weakly of P21 CYP1. The former can block the action of the Cyclin D CDK4/6 complexes, while the latter can block the actions of the remaining Cyclin CDK complexes. It means to say P15 INK4B can only work in the G1 phase, and the remainder as G2 and M phases. The P21 CYP1 can halt the brake or can apply the brakes to the cell cycle. So independent of this, damage to the cellular DNA causes a strong rapid increase in P21 CYP1, which in turn can shut down the cyclin CDK complexes that are active in the all the phases of the cell cycle after the cell has passed to the arc point in the late G1 phase. That was the video for today. We will meet in the next video. Till then, goodbye.